Okay, ladies. Last time we talked about how enzymes are things that speed up chemical reactions or control them, such as the breakdown of sugar in our body. And then we were looking at how different things affect enzymes. So one of the things we talked about was temperature. So I'm going to ask Kimberly Douglas, who's being recorded and will be going on YouTube, this question. According to this graph, you can see that the reaction rate goes up, and then you can see it goes down. So you can see it goes up on this side, and then it goes down on this side. Why does it go up initially?
It's working quickly.
Because as the temperature goes up, the particles move more quickly. Okay, very good. And then what happens is when it gets too hot for the enzyme, which is probably going to be a protein, when it gets too hot, the rate goes down because the enzyme starts to unravel a little bit. Its protein shape starts to change because the heat starts to affect the bonds in the protein. And then because remember the way an enzyme works because a substrate fits into what's called the active site. It's a perfect fit. So if heat starts to change the shape of the protein, the fit is no longer perfect. So you might get more collisions, but they might not be as effective. So the rate will go down. Okay, so this is what we talked about last time. So let's continue. So enzymes, talking about things that affect them, right? So we know temperature can affect an enzyme. Sometimes enzymes, no, not sometimes, often they need help. Okay, so anything that helps an enzyme is either going to be called a cofactor or a coenzyme. So a cofactor and a coenzyme are things that help enzymes work. So a cofactor is an inorganic substance that helps a protein or enzyme. So I'll give you an example. In hemoglobin, it's a protein that carries what special molecule? Starts with an O. Oxygen. It needs a very special cofactor for it to do this. What's the name of the cofactor that it needs to carry oxygen? And if you don't have enough of it, you could suffer from anemia. Iron. Very good. Okay? Iron. A coenzyme is similar to a cofactor. The difference is it's an organic molecule. Okay? So it's made up of lots of carbons and hydrogens and oxygens. So what would be the cofactor, if you remember we talked about this, that helps make collagen? Without it, you get scurvy. Vitamin C. Okay? So enzymes, we're talking about things that affect them. For them to be effective, they may need cofactors and coenzymes to work properly. Okay, let's carry on. Let's talk about things that can inhibit enzymes. So things that will have a negative impact on an enzyme. We're going to talk about two things, competitive and non-competitive inhibition. Okay? So you can see they're both inhibitors. They're both things that would reduce the reaction rate, not speed it up, but slow it down, or maybe even stop it all completely. So I'll show you a picture. So here's a picture of a competitive inhibitor, and there's one of a non-competitive inhibitor. Okay? Just by looking at it, the competitive inhibitor looks pretty logical. Why does a competitive inhibitor slow down or stop the reaction rate? Why? Look at it. What is it doing? Where is it going? What do we call this site here? where the substrate goes. It's called the active site. What do you notice about what it does? Where does it sit? It sits in the active site. So if an inhibitor sits in the active site, what's the problem? What's the problem for the substrate? So imagine this is the substrate. Can't get in. So how do you think a competitive inhibitor works? It blocks the active site. We'll study an example of this when we get to photosynthesis. We'll look at an enzyme called ribisco. It's over here. This is the enzyme that makes sugar, okay, for plants and other photosynthetic organisms. And these two molecules over here actually will compete for that enzyme. I'll give you another one. There is a molecule that can compete for hemoglobin's active site. Now, I just told you that hemoglobin wants to carry what molecule? Oxygen. There is another molecule that is made that can be dangerous. You have detectors in your house for it that will also competitively inhibit oxygen from getting to hemoglobin's active site. Carbon monoxide. Very good. Okay, now, how is it different than a non-competitive inhibitor? So, competitive inhibitors, you can see they're competing for the active site. The inhibitor and the substrate are competing for the active site. But if you look at non-competitive, how is it different? Where does that non-competitive inhibitor go? But where does it go, though? Does it go to the active site? No. See, you notice it sits into some other site, not the active site. So it's not competing with the substrate. But Emily, you notice something. You notice that when the inhibitor binds, what happens to the shape of the protein? Yeah, there's a shape change. And look what happens when the shape changes. The active site no longer is a good fit for what? For the substrate. So in both cases, whether it's direct competition or the inhibitor changes the shape of the protein, they both have a negative impact on the enzyme. Okay, so these are examples. I'll show you a neat little picture. This is an enzyme. Now, these three things here might not look the same, okay, because I pulled them off three different sites. This, and this, they're all the same enzyme, okay? Now, this one over here and this one over here use the same color. This one on this side uses a different color scheme, but all three are actually the same thing. It's an enzyme called HIV protease. This is the enzyme that builds or helps build HIV. Okay? This is the active site right in here. So here's the active site. This is the active site. So the substrate is this thing in magenta, this thing that's highlighted magenta over here. And you could see that this molecule over here, this one over here, and this one over here, these are called inhibitors. So here's the name of the inhibitor, right over here, and here's the other one. I don't really care if you know the names of the inhibitors, but I want you to know, I want you to be able to look at this and tell me, do you think this looks like it's competitive, or does this look like it's non-competitive? So remember, so the active site is right in the middle. You can see it's right in the middle here. And here's my substrate. Do the inhibitors look like they're going into the same area? Does it look like it's the exact same spot the substrate would go? What do you think? So take a look at this picture. You can probably see it really clearly on this one. So you see this section in red over here? Right here? You see the substrate is just right above it? Look over here. What do you notice? Right above it is where the inhibitor sits. So does it look like it's the same spot? What do you think? Guys, look right closely. See this highlighted section in red over here? You can see it right over here, this highlighted section. Okay. You see that this substrate is right above it? You see how this inhibitor is right, the inhibitor is right over here. You see how it's right above the section in red? Does it look like it's the same spot? It is. It's the same spot. You guys okay? Alright. So if it's targeting the same area as the substrate, then that has to be what? That has to be competitive. I'll show you a different example. Again, I don't really care if you know the names. Don't worry. I just want you to understand the difference between the two. Okay? So this on the left here, let me highlight this. This is an enzyme called CYP2C9. It's an enzyme found in your liver, involved in detoxification. And these molecules on this side over here, you can see. So if you were to zoom in a little bit, you might be able to see them a little bit better. There's a bunch of molecules that actually overlap here. Okay, they're a type of molecule called flavones. You don't need to know that. But they're all competitive inhibitors. Oops, sorry. Okay, they're all competitive inhibitors. Now, I want you to compare these guys on this side. So, these to this one. This molecule is called 6-hydroxyflavone. Tell me by looking at it, does it look like it's sitting in the same spot? It's hard to tell, but there's a little clue. Can you spot these two over here? I'm going to change the color, you might be able to see that a bit better. You spot these two things over here? This is called, you can see it says LEU. I'll make it a little bigger. LEU, and this is PHE. What do you think LEU and PHE are? What do you think this is? What is this whole thing? What is this? It's a protein. So what do you think LEU and PHE are? What are they? They're amino acids. Okay? And the number, I don't know if you can see the number, well, I kind of covered the number now, actually tells you the location of the amino acid. So you can see the amino acids are sitting here, and there's a gap between the inhibitors, which are sitting right over here, and these two amino acids. If you look over here, here's the amino acid, here's the amino acid over here. Here's leucine, here's phenylalanine. Look where this inhibitor sits. It sits right beside them. Does it look like it's sitting in the same spot? It's not. So this actually would be an example of non-competitive. Okay? So remember, the big idea is, if it sits in the same spot as the active site, what is it? It's competitive. If it sits somewhere else, non-competitive. Okay. Now, I'm going to show you something that may look like non-competitive, but it's not. I'm going to show you something called allosteric regulation. Okay? Now, I'm going to show you this picture here. And you're going to be like, this looks exactly the same as non-competitive, right? I see a molecule, and I see that it sits somewhere that's not the active site. Here's my active site over here, and I can see that if I take it away, the molecule, the enzyme, so here's my enzyme over here, I can see that my enzyme reverts back to its normal shape, and the substrate fits in. This diagram looks exactly the same as this bottom one over here, doesn't it? But this is not an example of non-competitive inhibition. This is an example allosteric regulation. Okay? They look the same, so it's confusing. They look very similar. Here's the difference though. This is something that a cell would do on purpose. Okay? So allosteric regulation behaves like non-competitive inhibition. Right? Behaves like it. Very similar in that you have a molecule that attaches itself not at the active site. And in fact, it attaches itself at an area called the allosteric site. So you can see it right over here. Okay, it's called the allosteric site. Now the reason why it's not the same is because it looks like it's the same, but allosteric regulation could actually switch the enzyme on. So you could have a molecule, you could have an enzyme that, so if you imagine here's my enzyme, I'll draw it like this. So here's my active site. So here's my allosteric site. I could have a molecule that attaches it and actually activates it. Makes it active, which is different than non-competitive inhibition. Because in non-competitive inhibition, when the molecule attaches, the enzyme is always switched off. In allosteric regulation, the molecule attaches and can be switched on or off. It depends on what the regulator is doing. Here, what's going on? This regulator is an inhibitor. You can see it right here. It's actually labeled inhibitor. Because you can see when it's bound, what happens? The enzyme can't fit the substrate. So if a regulator was an activator, and you'll see examples in this course when we get to that section, and we'll get to it, an example of this in genetics. Well, if the regulator is an activator, then when it attaches, it actually turns the enzyme on. Okay? So it's not the same as non-competitive. It looks similar, but it's not exactly the same. And because we have the word regulation, this is actually really important. Okay? Regulation. What does it mean if you want to regulate something? What's another, like a synonym for regulation? Give me another word for regulation. What would be another word for regulation? How about control? If something's regulated, that means it's controlled, right? In society, what's regulated? Drugs, right? Smoking to minors. A lot of things are regulated. Control. Another word, think of regulation as control. So allosteric regulation is a form of control. Okay? It's different than competitive and non-competitive inhibition. These are things that the cell has no control over. You have no control over if there's carbon monoxide in the environment. What you do have control over is moving away from the environment, right? But you can't control if the carbon monoxide binds to the hemoglobin. Your body does not, has no control over that. So, in regulation, you do. This is where your cells actually control enzymes through the binding of molecules to this allosteric site. What that does is it turns them on or off. So this is something that the cells do on purpose. So I'll give you an example here, and we're going to study this one in the next chapter. There's a really important enzyme called phosphofructokinase. Now, what does this enzyme work on? What do you think it works on? Fructose. And it's involved in making energy. And what it says here is that this enzyme is switched off when you have high levels of ATP. Now, if you remember, what does cellular respiration, which I'll call CR, what does it give you? What does it make? It makes energy in the form of ATP. This enzyme over here is a very important enzyme in cellular respiration. So let me ask you a really simple question. What if you have enough energy? Do you want to keep making more of it? What do you do when your cell phone battery is fully charged? You leave it in the wall? What do? You unplug it, right? So, cells have to have a way to switch off things. When you have enough energy, maybe you want to take that sugar and move it somewhere else. Maybe you want to store it or convert it into something else. So high levels of ATP will actually attach to this enzyme and turn it off. Okay? It's a form of control. It's really important that cells control things so that they know when to make it, when to switch something on, and when to switch it off. And I'll show you an example of allosteric regulation. That's also, this is called feedback inhibition, and it's a form of allosteric regulation. Okay? Now, in feedback inhibition, the idea is really simple. It's basically the same idea I just explained to you. Your body wants to make something. So in this case, it's not energy. In this case, we're making a molecule called isoleucine, which is an amino acid. Okay? Your body has the ability to take this amino acid over here, and you can see convert it through a series of steps into isoleucine. So let me start with a really simple question. When would you want to make isoleucine? A, when you have lots of it. B, when you have very little of it. When would you want to make it? B, correct answer. So it's B. And I'll draw you a little smiley face. So what happens is you have a bunch of enzymes, and here only one of the enzymes is labeled, but there will be 5 of them. So what happens is this enzyme over here takes threonine and converts it to this molecule. And then this enzyme over here takes this molecule, alpha ketobutyrate, and converts it into this molecule. And then so on until you get to isoleucine. Well, what happens when you have enough of it? What do you want to do? You want to turn it off. You want to have a way to switch it off. So we can imagine that this enzyme, threonine deaminase, works on threonine. So I'm going to draw threonine as a little rectangle. So you can see right now that it fits. What happens though is when you make enough isoleucine, the isoleucine will actually attach itself to that allosteric site, which I'll call AS. And by attaching it to that allosteric site, we'll probably change the shape of this enzyme. Sorry about the drawing, it's really bad. But here's my isoleucine right over here, changing the shape of my enzyme so that the active site no longer can fit the substrate. By doing this, the enzyme is switched off. And if you switch off the first enzyme, you can't make this, which means this enzyme can't make this, which means you can't make this, which means you can't make this, which means you don't make this, which means this whole thing is shut what? Down. Until you run out of what? Until you run out of isoleucine. Once you run out of isoleucine, this gets knocked off, the enzyme reverts back to its original shape, threonine can fit in again, and the process begins again. Okay? So these are just things to consider when we're talking about enzymes, things that affect them, things that regulate them, and that is the end. Let's turn this on.